This is going to be a quick review on the topics that are in Unit 3. And for this, I have already written on the announcements things that are important. And what I would like you to do is not only read all your notes thoroughly and listen to the narrations, but also, when you observe these PowerPoint images as you see here, every little image will give you a good clue and will help you in your test. For example, if you look at this first image, which is the beginning of the cerebellum, notice something like climbing fibers from the inferior olivary nucleus. So you can see that from the inferior olivary nucleus, those fibers are known as climbing fibers. Everything else is called mossy fibers. Also notice the connections, and these are something that you've already done. The inferior cerebellar peduncle is from the medulla, the middle cerebellar peduncle is from the pons, and then the superior cerebellar peduncle is from the midbrain. So these are little things which each image will help you with. And then when you look at the rest, always know the gross features of any structure that you're looking at, be it the cerebellum, the thalamus, the internal capsule, the basal ganglia, for the visual pathway, the eye. It's very, very important. So up here, you must know about the dorsal surface, the ventral surface. Then you're looking at the side view. Notice how the cerebellum is sitting on top of the fourth ventricle, so it forms the roof. And up here also notice that this dentate nucleus is present within the core of the white matter. So it's not in the cortex, but it's present in the white matter. So these little things will always help you. Then, of course, you must know the connections. So these are the different parts. And then for the connections, if we skip to this part here, so here you're looking at the cerebellar afferents. So you must know which afferents are passing through the inferior peduncle, the middle peduncle, and the superior peduncle. And then when you go to the next one, you notice the efferents. So here you can see that the middle cerebellar peduncle does not have any efferents. So that tells you that the superior and inferior have both afferents and efferents, and the middle one only has afferents. Little things like this will help you. And then let's go back to this one here where you're looking at the internal structure. So here you're looking at the cortex. So all of this is the cortex of the cerebellum. So notice how you have these different cells in the cortex, and of these different cells, remember the Purkinje cells, which are these were most important. So you can see that it's lying in the cortex, whereas the nuclei are the ones which are lying in the white matter. So these Purkinje cells lie in the gray matter of the cortex, not in the white matter. So if a question comes, the Purkinje cells are seen in the white matter, then you know that that is a wrong statement. So little things like this are extremely important. And then of course when you come down here, these functions, these are very important. So I won't go through this, but you must, knowing all these functions will help you to answer questions in the in the clinical situations. These again are connections. And then when you come down here and you look at the signs of cerebellar dysfunction, and I kind of tell you that how there is a resistance to movement normally. So when that resistance goes, you see hypotonia, as in cerebellar dysfunction. So a question could be given based on something like this. You're picking up a child and you notice there is no resistance, so this is a sign of, and you can see cerebellar dysfunction. Another up here again, this image is showing you how a child with cerebellar dysfunction, to keep her balance, she walks on a wide base with feet wide apart. So that's why it's very, very important to know and look at these images very carefully. The other thing is these intention tremors. Notice how these tremors increase as the person comes, if they're doing something as you near the object, like for example the finger nose test or other things that I've mentioned. So again, very important to be looking at that. And here again you have these same thing, the finger nose test, and notice how this test for ataxia, where you hold a toy in front of a child and you ask the child to touch it, the child is not able to touch it, and so that's important. You must know these little terms like this. What is dysdiadochokinesis? What dysarthria? What kind of speech is seen? So these are some of the important things that you must know. So that's why I'm kind of asking you to pay attention on all of these different aspects of the cerebellum. Also know that the cerebellar control is ipsilateral. Know that the cerebellum does not have any direct contact with the anterior horn cells. Remember, it influences it indirectly. So if anything special like that is mentioned, you must know that. In relation to the thalamus, again, the nuclei which I told you, which were important, your VPL, VPM nucleus, you must know what tracks end in the VPM and VPL nucleus. Those were very important. Then you have these lateral and medial geniculate nuclei, again, what they are associated with, so that is important. Then remember that what the thalamus is related to, and I think the next the next few pictures probably give us that. So here you can see the thalamus, how this part here will be the lateral ventricle, and when you come here, again, notice these relations of the thalamus. Notice how it's related, its medial surface is related to the third ventricle, whereas it forms the floor of the lateral ventricle. And then notice the relations of the internal capsule. So when you're doing the internal capsule, again, you look at the same image. So it's important to know the different parts of the internal capsule and which part is related to which area of these nuclei. And then up here, of course, you have these different connections, and I've identified the important ones. These are again connections, so you must know about these. And then lastly, these are the thalamic peduncles, where you need to know which peduncle is connected to which area, and in the area also I give you the functional areas. So a question could be based on that. The visual cortex is connected by means of, so then the posterior thalamic peduncle. So you must know these two. And then in the reticular formation, this was just general, so little things that I've mentioned on how it controls the cortex and arouses it, and how general anesthetics act on it. So these are little things that you must know. So all of these functions in the reticular formation. Then when we go to the visual system, and let's say you start from here, the eyeball and the retina, you must know the different layers. And then how I mentioned about retinal detachment and what these axons are continuations of. Then up here, this is showing you the different layers. Notice how the chiasma is formed, how it is so close to the pituitary gland which is present here. So in the lesions, you notice if the pituitary gland is enlarged, how it will affect the chiasma. So that's why it's very important to know these relations in any of these structures. Then the visual pathway, I cannot emphasize how important it is. So you must know how it goes from the retina, goes all the way down. I'm not going to go over that, but you must know this. Remember, it's the field of vision. So if you look up here, it's the left field of vision, and it goes to the right visual cortex. And then here, these are little important points in the visual pathway. And then the reflexes, again, these are very, very important. So you must know the pupillary light reflex. Notice how it goes, and how this pupillary light reflex, how it goes to the pretectal area, and then from the pretectal area it goes all the way down, and it goes to the sphincter pupillary muscle. And then you have that consensual part where it's going to both pretectal nuclei. And then we have the pupillary dilatation reflex. Again, you must know this, and how its pathway goes all the way to the dilator pupillae. And then the last one, the accommodation reflex. And notice how the accommodation reflex, it bypasses this pretectal nucleus. So that's why in Argyll-Robertson pupil, where this pretectal nucleus is damaged, the accommodation reflex persists, whereas the light reflex is gone. So again, little things like that you must know in this. So wherever these important clinical applications are, I cannot emphasize the importance. Again, you must know the features of papilledema. And then in this lesions of visual pathway, these terms you must know: hemianopia, which is loss of one half of the field of vision. Homonymous is when the same half, that is the right or the left half, is gone. And heteronymous is when one side is right, one side is left. As for example, the bitemporal is referred to also as heteronymous hemianopia. And then, of course, these lesions, like I said, the Argyll-Robertson pupil, you must know the Horner syndrome, and then, the brainstem injuries. So again, extremely important to know these little things. So if you kind of go through your PowerPoint and look at these, and then with your notes alongside, it'll really help you. Then when we look at the basal ganglia, and with the other areas, so let's go all the way up to the top. So the components of the basal ganglia, again, look at these very carefully. Notice where each of these structures are present: the caudate nucleus, the amygdaloid. Notice again how it goes up to the temporal lobe. Then you look at the lentiform nucleus up here in the lentiform. You must know. So read your notes very, very carefully. You must know the difference between corpus striatum and striatum. So that's, I've given that in your notes. Notice where the substantia nigra is. So these little things are important. So gross features and relations, very important. And of course, you must know these functions. So these little functions, and write out that. Notice how it prevents unwanted movements, but also causes stereotyped activity, gives motor expression to emotion. So these are important. Then you should notice the position of these nuclei in relation to the internal capsule. So when you're doing the internal capsule, you must know the parts of the internal capsule and how they are related to the different nuclei. Then these clinical correlations with all these diseases, you must know these diseases, and that's why if you look at those videos, it'll really help you. So all of this is kind of just straightforward. So you must know all the different things that you see in these diseases, what these diseases are caused by, in other words, which nucleus or which part is affected, or what neurotransmitter is lacking. So very important to know all of these. And then, of course, for the internal capsule, again, know the different parts of the internal capsule, how it is related to the nuclei. For example, the lentiform is lateral to the nuclei, the caudate and thalamus are medial. Know the different parts of the internal capsule. And then also the blood supply of the internal capsule is important. And when you're doing the different parts, you must know which part contains what fibers. For example, the corticonuclear fibers are present in the genu, the corticospinal, and of course, the sensory pathway is present in the posterior limb. So that's the part which is related to the thalamus. And then, of course, the blood supply of the internal capsule, extremely important. So you must know that. And then any particular artery which is important, which I mentioned, you must know about that. So these are some of the things that are really important. So it's basically whatever I've given you in your, in your announcement, but I'm just going over these little parts here to kind of emphasize this.